Okay, thank you. So, good evening, all of you, and thanks, Sharma sir, for introducing me. So today's topic is understanding India's small mammals. There's the identification, methodology, and conservation. So, so today's talk that highlights are that how we know about the mammals, then that's methodology of collection and preservation, then role of taxonomy in identification of small mammals, then study of small mammals through non-invasive methods like that owl pellet analysis and all, and lastly on that conservation implication for small mammals. So what are the mammals that mammals possess? Just like we are that beings are also mammals, and other smaller mammals also that like shrews and all these all are under category of class Mammalia. So they possess that mammary or milk producing glands for nourishing their young ones, and then possession of true hairs and sweat glands on their body. Even that in pangolins also, that juvenile pangolins having the true hairs on, in between that scutes and all. Then possession of different types of teeth as per feeding habits, that's carnivores having different than articulans, that omnivorous that shrew having different, then rodents having different types of teeth. Then possession of link between that mother and fetus, that placenta is available in mammals. Then they are warm-blooded animals. They adjust their that temperature as per the environment and all. Then having that parental care of that young ones. So that by definition, the smaller size mammals, medium size, and these are if one category is there. Less than 30 centimeters are that small size mammals, but uh, most of that rodent, that's one that porcupine having that larger than 30 centimeters. But 90% that rodents are that smaller than the 30 centimeters. So all that rodents are under the small size that mammals. Then medium-sized mammals that between that 30 centimeter to 100 centimeters, like cats and all, dogs, and large-sized mammals that head and body length above 100 centimeters, like tiger and elephant and all these. So the phrase that small mammals does not constitute a clearly defined taxonomic entity and is generally considered to refer to any assemblage of mammal species with individuals head and body length less than 30 centimeter and that live weight do not exceed 5 kg. So in adult stage. So in pictures, see this Suncus etruscus. This is the smallest mammals, terrestrial mammals of the world. This is a pygmy shrew, and other groups are under small mammal category, that is Ellipotaphala. Under that, the shrews, moles, and hedgehogs are there. Then Scandentia, tree shrew. Then Rodentia, mice, rats, gerbiles, bandicoots, squirrels, porcupines. Then Lagomorpha, the hare and pikas are there. And in under Chiroptera, we study that bats. So, without collection of the any single specimen, we can't identify up to that species or subspecies level of that species. So, needs specimens from the field or that museums. So, need the fresh specimens for the field. Needs different types of tools for rodents and that shrews, like that wonder trap or that wooden trap, then Sherman traps. This is foldable trap, aluminum one or steel one, and then the snake break back trap we use in houses. But this is not used in taxonomy because that head will struck and that skull will broken. So these live traps are very much important for taxonomic studies. Then that glue trap is also there. The glue trap use that some sticky material on that, and that animal going through that route, so here that will stuck on that greasy the chemical or that component, and will get that specimen. But for permanent that preservation, that is, hairs will be lost or so much sticky, but we will get that skull and other measurements also. Then for bat collection, we need that mist nets that collect live bat specimen from foraging area. And insect net we use in bat collection at a roosting site, just like the cave or that old forts or tombs and all. So when you get that specimens from that field, we that euthanize that specimen by using the diethyl ether or chloroform, this euthanizing agents, and will kill that animal and preserve for the further process. So before after euthanizing the specimen, we will take the fresh measurements, because after preservation that will that specimen will be shrink and all, will not get that proper that measurement. So in the field, whenever that euthanize the specimen, so we will take the fresh that measurement. In rodents and shrews, we'll take that head and body length, and then that is nose to anus, and the tail length is nose to the last vertebra, and then hind foot length and ear pinna. In bats, different that measurements are there, including head and body length and tail length. Some that this forearm length is very much important in taxonomy of that bat for identification up to species level. And other measurements are that third metatarsal, the second to fifth metatarsals are there. So we'll take that other measurements also, and ear pinna and tail length. For this larger or medium sized mammals needs that height and girth at various points are recorded. But we are talking about the small mammals, so we'll take measurements of these phalanges and all, or the metacarpals. So come to the preservation part. So that preservation includes two processes: this fixation and that preservation. So in fixation, will we use that 10% formaldehyde? That formaldehyde is already a 40%. So we'll take that one part of formaldehyde solution and three parts of water, and that the body will fix by this 10% formaldehyde. Uh, Today's world is doing that barcoding and all DNA barcoding. So before the fixation in the formaldehyde, we'll take the extract the tissue from the body, that the specimen, and preserve in 70% or absolute alcohol for DNA barcode of particular specimen and label the vial. Uh, Nowadays, this formalin or fixation process also drop. Some people, whenever that alcohol is not available, so the people will that preserved in that formaldehyde. But if alcohol is there, so directly we will take the 70 to 90% depend upon the body size. Will that preserve directly to the 70 to 90% alcohol. Uh, the benefit is that anytime you will take the that tissue extract in the laboratory also, after coming to the laboratory from the field. So this preservation of mammals, the fixative, the 10% solution that injected in the body through that incision taken in the ventral side, that abdomen near to abdomen, and insert that by syringe or that fixative material. So that specimen will be properly fixed or properly preserved. The preserved material, the specimens will be then shifted to a jar and with label and all these things, and keep in the our museums for further studies for taxonomic identification. So this is the earlier this wet preservation, and now this dry preservation. In dry preservation, the skin of the animal is removed and treated with that chemicals to fix the tissue. And that dry preserve is useful for preserved medium or large size mammals. Just like if that cat specimen is there, to remove all viscera and take that skin and preserve properly in for museum exhibits. But in the smaller mammals, preparation, two types of that processes are there: the skinning and rolling. So this is very tedious process and will take time and all. Also, to understand this process needs the hands-on training and all these things. Not in detail. I'm not going on details in today's talk. But this is process is very lengthy, and that different type of chemicals and all used for that. This animal first skin in a process similar to removing the skin. That skin is animal that cut with a scalpel longitudinally along the middle of the abdomen for about 2 to 3 centimeter, and care was taken not to open the abdominal cavity. The skin was separated all around up to the base of the tail. That magnesium carbonate was sprinkled on the inner surface of the skin to facilitate gripping. The skin was held at the base of the tail by the left thumb and forefinger, and the tail vertebra were pulled out of the skin by the right hand. Then freeing of the skin. This is all the process of that skinning of that small mammal specimen. And after this skin will that viscera are taken out, the skin will applied with different type of chemicals like magnesium carbonate, potash alum, then borax, synthetic pyrethroids. Earlier we used that arsenic soap. This composed of that camphor and arsenic oxide, carbonate of potash, soap, and lime powder. But that arsenic is carcinogenic, so then nowadays we not use that arsenic soap. Instead of that, we use that synthetic pyrethroids or borax and all. So then large scale in the place in the salt and alum water also. So the gripping of that hairs and all, and that we remove the, after drying, that remove that fat, because this the fat, if fat is there, that skin will damage, or that insects will damage that skin also. So scraping the dry skin to remove the fat is very much important in the taxidermic process. So this is rolling. That roll specimen for further identification, because this traditional method is that rolling is by filling inside the cotton wool. The viscera is not there, only the skin is there, and that cotton wool or sawdust inside there. And the shape of rolled specimen, you have to keep the specimens in that boxes and all these things, and you tag that label and all. So this is easy to manage that specimens to further that fumigation and all these things for further use. So after skin, that skull will remove or that from the skin, and during that process of that skinning and all, the skull will cut from the viscera and boiled in the water, and or if that skull is very sturdy and muscles is very sturdy, then will put that some KOH pellets and all in the boiling water, and depend upon the size of that skull, will take half an hour, 15 minutes, or different temperatures, and then removed all that muscular part from the, and then that skull will clean by forceps and while that scalpels and all. Then that skull will tied with the skin, with kept separately in the glass vial for further reference. And for if you want to prepare the skeleton, so you want to bury that specimen under soil. That will take time for 2 3 that months or 6 months also, depend upon the size of that animal, and you will prepare the skeleton also. But the skeleton is not important for identification, because the skull characters and that morphometric body characters are very much important, and we will identify up to subspecies level, no point. So after removal of that muscles and all that, the skull is prepared, and we'll take different type of measurements like occipital nasal length, that condylobasal length, then that nasal length, then interorbital length, hygomatic length, then anterior palatal foramina, then palatal length, these molar to throat length. These all are very much required for the identification of the species or subspecies level, because that keys are older, and that giving that proportion of that species having that characters. So this occipital nasal character is very much important. After that, these characters given in the proportion. If you follow the keys of Ellerman and all that, so identification that the percentage of that molar tooth row with occipital-nasal length, that percentage of bully, tympanic bully with that occipital length. So these type of keys are there. So for that measurements will take that should take that key properly. So we'll go to the species level identification. So this type of data sheet we will use, having all the details. This a morphometric character, that head and body length, tail length, that hind foot length, and ear pinna length, and that after that the craniodental length, cranial, that occipital nasal length, condylobasal, that nasal, and all dental, that molar tooth row length. So these all measurements will keep in the data sheet, and after that we'll take that that head and body with that proportion with tail length percentage, then head and body with hind foot length, then ear length, these all percentage and all we write in the data sheet, and then we follow the dichotomous key for identification of that specimen. So this labeling is also very much without label, that specimens don't have without no importance to that specimen. So this label is tagged with that specimens having that locality date, then date of collection, then name of collector, measurements, because that field measurements we will write on that back side of the that label, that will help to the next researcher also, whenever he will study that specimen, will get that that field measurements are there, that that will use for his study. Then additional data like that collection number, or then field number, or then number and position of mammal lactating or not, reproductive conditions of gonad, number of fetuses. These all according to your study, you will take that data on that label. Then in rodents or that smaller mammals, this mammary position and that number of mammary glands are very much important, and their position. Just like this is thoracic, abdominal, inguinal. So this is a Rattus rupescens. This is a house rat having that 5 pairs of that mammae. So in some species having 3 pairs of mammae, 4 pairs of mammae. So then in the Bandicoota bengalensis, that more than 6, 7, 8 pairs of mammae. Bandicoota indica having 6 pairs. So this type of that's number of mammae is countable in the female specimen, and that will help for identification of the species. These plantar pads are also very much important of hind foot. So see the picture, this Bandicoota bengalensis and other ratters and all this having that 6 plantar pads in the hind foot. See the Millardia meltada having 5 plantar pads. So this is also one of the important character. You will identify in the field also. If this 5 is there, so we'll go through that Millardia meltada is there. So this is very much important character for identification of the smaller mammal species, especially in the rodents. So maintenance of museum specimens by that fumigation of insecticide, then keep PD benzene, that powder in cloth pouches and in showcases. So when that smaller insect will damage that skins and all, so needs these pesticides for for pesticides that through that if that damaged skins are there, so will that our that all the uh, process will not use it. So for that these all PD benzene depend upon the size of the animals or the dry species, the dry that skins. You use that PD benzene or this naphthalene ball also or camphor in specimen boxes. Time to time you will manage or uh, check that insecticides are there or not. To change the naphthalene balls or camphor repeatedly. So how that museum specimens are important in that taxonomic studies. These are the reference collection and allow direct morphological and skull comparisons across historical populations. So see this our collection having this Pune station uh, started on the 1960s. So 60 years that specimens in our museum. If anybody study that specimen, so they will get that morphological and skull comparison data on from that museum specimens. Then also served as accessible that DNA sources for phylogenetic and conservation genetic studies. So if that specimens preserved in that alcohol or some earlier specimens are arsenic preserved dry skins, so that will not more helpful because the DNA will damage and not will get that percentage of basis. So that alcohol preserved specimens will get that DNA from the tissue. Then museum specimens will help in documentation of that morphological variations, because hundreds of if we study that hundreds of specimens having different measurements and different variations. So through that multivariate analysis, we will identify the key characters and all. So that is also helpful for that study. And then specimen labeled with that locality data, that geographic information or the coordinates that through take the time or GPS data, with that helps in preparation of species distribution maps and that geospatial analysis that will carried out through that GIS tools. So only if the earlier specimens not that return that coordinates, but from that locality details. At present, we will take that from our Google Earth or will take that coordinates, and that coordinates will help for the distribution maps. So that role of the taxonomy in identification, that after morphological identification by the physical characteristics like body size or that village color or fur color or cranial features to separate species in the field or in the lab, the standardized neighbor that naming is required. If I say that that field mouse, so I say I know that field mouse, but that layman also in other countries that field mouse is a different one. In our countries that field mouse is that masbuduga. That for that that standardization is required. For that replaces confusing local common names with a universal and standardized binocular nomenclature. So ensuring clear communication among the global researchers. For that standardization, we will use that binomial nomenclature, and also for subspecies level that trinomial nomenclature we use. Then that dichotomous keys, that taxonomy studies provide the diagnostic keys used in the field or lab. This allows researchers to identify species by analyzing specific morphological traits such as the skull measurements, fur color, tail length, and dental patterns. So this is very easy. The dichotomous through that dichotomous keys, we will identify the species up to species or subspecies level. This taxonomy is also important in resolving that cryptic species. So that smaller mammals often look identical, but belong to entirely different species, like that cryptic species. So that in modern technology utilizes that genetic approaches like DNA barcoding to reveal hidden evolutionary differences that physical traits cannot distinguish. So, just example of Mus buduga, he is looking similar the of species that Mus terricolor. Earlier, that terricolor synonymized under Mus buduga. Nowadays, through that molecular studies, this Mus terricolor is a valid species other than that Mus buduga. So this DNA barcoding is very much important for the taxonomy or identification of the species. For that conservation and management also, this identifying species, that exact taxonomic unit is critical for prioritizing conservation. If you know that species is there or that area or protected area, if the species is endemic for that reason, so you will prioritize that conservation or managing ecosystem that will help to that managers for conservation of the species or conservation of the habitat. Some identification taxonomy in nowadays in bats, that acoustics, bioacoustics are used. So especially vital for that identifying ecology, echolocating that small mammals like bats, where calls are analyzed against established taxonomic databases. So without harm to any specimens and all these things. So we'll take the through that bioacoustics, we'll identify up to species level. So identification of mammals, that a mammalia, class Mammalia is divided into three subclasses, that monotremes, that marsupials and eutherians. So that monotremes, that egg-laying mammals, then marsupials, that host mammals. These both are not in Indian, in India, found in India. Only that eutherian, that placental mammals found in India. So they are represented by 13 orders and around 459 species reported from that mammals reported from India. This is according to that checklist of mammals last year published. Uh, uh, this year not published, but this is under preparation. Added three more species. One that shrew species from Nagaland, that Nagasorex albidens, and that two that bat species newly reported from northeast area. So these are this mammalian diversity, that profile. So the 13 orders they have. That Proboscidea, this elephant is one species. Then Sirenians, this sea cow, one species. Then Scandentia, tree shrew group, having 3 species. Then primate, 23. Rodents, 111 species. Lagomorphs, that hare and pikas, are 12. Then Ellipotaphala, or that all insectivores, 37 species. One in now added, that Nagasaurus, 38 species. Then Chiroptera, bats are 135. Now this 137 or 138. Then Pholidota, these pangolins, 2 species. Carnivores are 59. Then Perissodactyla, or that even toad, 3. Then Artiodactyl, 37. Then Cetartiodactyla, that marine mammals, 35 species. So total, as per that our mammal checklist 2025, that total 459 mammals found in India. So in a smaller mammal category, that only we'll talk about today in today's talk, these Scandentia, Rodentia, Chiroptera, and Elliptotaphala group. So this is the Scandentia, this treesure group. In India, 3 species are reported, that Tupia belangri in northeast area, then Tupia, that Nicobarica, this endemic to Nicobar region, and one is this Anathena eloti, this is reported from peninsular India, from down to Ganga, and this is endemic yeah, to India. And this looking very much nice group. This snout and rounded ear like a typical shrew, and that bushy tail like a squirrel, and the body form like a mongoose. So whenever the crawling on that going through the roads, we will that confused with this smaller that or juvenile of that mongoose. So this is very much nice group, that Scandentia. Then the rodents, 111 species of rodents reported from India. So these classified into three suborders like that Suromorpha having that squirrels, then Myomorpha that rats, mice, voles and gerbils, and in Mysticomorpha that porcupines are there. This, if please see this area of that between the incisors to the first molar, upper molar, having the gap, that's called a diastema. This only found in the rodents and the lagomars, means hare and picas. So these character only found in rodents. So these are the rodent families and their distribution. See, the seven families are reported from India. This Suridae, having that squirrels and marmots, their distribution is throughout India. The marmots only found in Himalayan range. This Dipodidae, that birch mice, only one species is there. That's Cysista concolor, reported from Kashmir, Ladakh, and Himachal Pradesh. And Platacanthomidae, that spiny tree mouse, reported from Western Ghats of that Karnataka, Kerala, Tamil Nadu area. And then bamboo rat, there under the Sparacidae, only two species are there. This from Northeast India. Then chrysitids, that holes and hamsters, reported from that Jammu Kashmir and Western Himalayas and Northeast India. And that larger group is Muridae, covers that gerbils, mice, rats, bandicoots, and that distribution is throughout India. And Histidicidi, that porcupines, also reported from India, throughout India. So see these pictures. These are the color morphs of that giant squirrel, that's Ratufa. So this Ratufa indica is reported from northern part of Western Ghats to the southern Western Ghats. In see this Bhima Shankar and all, the Konkan, Goa, then up to the Wayanad, having this Ratufa indica. This tail tip is very much that white or yellowish color, and if you go to the Bhimashankar area, that Ratufa indica, Elbestoni, now that cinema is under Ratufa indica, having tail tip, tail that two third of the half, more than half are whitish in color. So these are color forms, and then that Ratufa indica maxima, this is wholly black in color, and the tail is also black. This reported from the southern Western Ghats area, that Tamil Nadu, that Kerala, Coorg, and all the Tamil Nadu part of area. And this Ratufa indica, that central is this uh, distinct black shoulder, that pattern is there. So this is a distribution in central and the eastern India up to that Odisha and all. So this is very much interesting group, and see, this is the difference between this is this grizzled giant squirrel, Ratupa micrura, reported from that southern Western Ghats, Karnataka, Tamil Nadu, and Kerala area. So the see the skull pattern, or this is a upper maxilla, and see this palate size and this palate size is very much different. Also the shape of this bony part is also different. So up to this skull is also very much important for the taxonomic purposes. Uh, if that giant squirrel, you not shoot that giant squirrel and all these things. Now the taxonomy is stable in the giant squirrels. So but this skull characters is very much important for identification. Oh, these are the striped squirrels. Uh, Here also very much different forms are there, region to region. This Funambulus Funambulus pennanti Chhattisgarhi, Funambulus pennanti, that lutiscence and all. For different regions, shape and sizes, but our study found that Maharashtra population is all throughout Funambulus penanti, and that Chhattisgarh earlier reported from that Nagpur area and all. These are all synonymized under that Funambulus penanti. So, and in Funambulus palmarum, that three-striped squirrels having also a number of forms or that subspecies. There's Funambulus palmarum from that southern area to up to Bihar area, then up to Andhra Pradesh and all. Then Funambulus palmarum robertsoni up to that Bihar and central India part. Then Funambulus palmarum bellaricus up to that Deccan Karnataka part. So this type of forms are there. Depend upon the skull size and then color of that specimens and tail length and all this. So these are the scope in further scope for if molecular studies will do. Maybe that the forms will be resistor to up to that species level also, or the synonymized in the earlier that Funambulus palmarum. So these are the this bandicoot rat, a big size, the tail short and all, and this is smaller size, that little Indian field mouse, a tail bicolor. That upper part is dark and lower part is pale, and in mus, number of species are there: mus buduga, mus saxicula, mus platatris, mus philistris. So by photographs, I could identify. People will say that will identify that for confirmation. They send that photographs of this mouse species. So I couldn't identify on the basis of that photograph, because the skull characters are very much different. You have the skull, that molar tooth of that mouse buduga, mouse that Phillips having 3.2 or 3.3 mm, and in months mouse saxicula and platypus having that 4 plus mm, 4.5 or near to 5 mm also. This is Lagomorpha group. That 12 species are reported from India, and they are closely related with the rodent, except the structure and number of the teeth and the structure of ears. There are four incisors in the upper jaw. See, these two pairs of incisors are reported. See in this arrow mark. In rodents, these lower only two pairs of incisors are there, and that ears are larger in size in the lagomar. In these rodents having smaller size. So these are the hedgehogs. Three species of hedgehogs are reported from India. These Hemicanus polaris, this northern India, this Gujarat and Rajasthan part, and this Paraicanus micropus, western northwest India, and now it's reported from our Maharashtra area, from Nandurbar, Dhule, and up to Jalgaon. People are reporting this is of. And that third species is that Paraicanus, that nudiventris, only reported from Kerala, Tamil Nadu, part of Kerala, and part of that Andhra Pradesh. This family Soricidae under that Illipotapla. See, this skull part, these are without that zygomatic arch. So this is a very much a distinguished character of these rodents having this the zygomatic arch. But this Soricidae family of the Illipotapla, the shrews, that Sancus murinus are all having doesn't having that zygomatic arches. And in molar tooth row, see that no gap is there, just like that rodents, that diastema. All are their incisors, then that premolars, molar, continuous that teeth are there. And vibration on that tail, on this snout is very pointed, long and pointed. So these shrews are 32, about 32 species are reported from India. This another group of Lepidoptera, this Talpidae or moles. Two species are reported from India, and this from northeast India. That Euroscapter microrus and Parascopter lepres. So these body are elongated and cylinder. This heavy that see these claws are very much heavy to for burrowing the soil, and this no external ear pinna is there, and eyes are very small and reduced because these animals most of the times lives in the burrows. So very much group that larger size group is 135 plus now that two species added bats. The flying mammals possessing that membranous wings and between elongated that second to fifth of these digits all are connected with the patagium, and they are capable for sustained flight with the help of echolocation system. So see, this is frugivorous bat, and this see this curl pattern and dentition pattern in this insectivorous bat and that frugivorous bat. The frugivorous bat having that dentition is very much simpler form, but in that insectivorous bat having very much complicated or very sharp and all different types of that dentition is there. These Pteropodidae or fruit-eating bats, the 12 species reported from India. And this, all there's a Pteropus medius on the this diurnal roost on the big trees or ficus and all. And some species that Rogetus listenalti are reported in some caves or that forts and all. Thousands of that individuals lived in that caves and all. See, Rhinolophidae family is having 18 species, and see, distinguished with that horseshoe bat. See, this horseshoe shape of nose leaf, the base of nose leaf is there, so called that horseshoe bats. So this horseshoe, then sela and lancet, and the different size in different species and different characters are there. Other is that leaf nose bat, hip family, Hippoceridae. So having that supplementary leaflets and others, the structure is very much different of that having anterior portion, longitudinal reeds and median portion and posterior. This is the Hippoceridae lankadiva, having that four that supplementary leaflets are there. So we'll count these one, two, three, four, and we'll take identification up to species level. These Falsemper bats are two species reported from India. That's Megaderma spasma and Lyroderma lyra. The two differences are there. To see the nose leaf having that end is that truncated, and here is not truncated, and the base is a heart shape is not present that heart shape. So these both species are distinguished, and this Megaderma spasma mostly reported from the forested area, and this Lyroderma lyra reported from that urban, and there's the old dilapidated that buildings and all. These mouse-tailed bats, two species reported from India, that's Rhinopoma hardwicki and Rhinopoma microphyllum. And most these species is reported in the semi-arid area. So this tail is longer than forearm length in hardwicki, and tail is shorter than forearm in this microphyllum. So another group is the sheet-tailed bat, the family under Emelonuridae, and these six species are reported from India. The tail is loosely enclosed in that interfemoral membrane, and the tip projects out from the upper surface of the membrane. And the gular sac is present or absent in the species. This is Tephajus melapogon, and this is male specimen having beard-like structures. This is black, so this is easily identifiable in the field. This male Tephajus melapogon. Another is that family Molosidae, that three-tailed bat, having that wing attached to the ankle and tuft of hairs on the dorsal side of the hind feet, and its tail is very strong and projecting for beyond 3 age of interfemoral that membrane. So these four species reported from India. See, this is largest group in evening bat. 63 species are reported from India. Uh, these mostly moving or that in the foraging areas. It's a evening, so they called evening bat. Very smaller size and not identify in the field. So needs at least one specimen for identification from that if a roost is there. So at least one specimen will take sacrifice and identify up to species level. So that will helpful for that roost and all will they then conserve that area if that species is endemic or other than that disturb that area after identification of the species. Then that long-fingered bat, that three species reported from India, and this highly developed second phalanx of third finger is approximately three times the length of the first phalanx. So earlier, these our area species, this reported from Roberts Cave and all this area in that Mahabaleshwar. So earlier that called as that Minipterus phylogenosus, but now that on the, through that molecular studies, these identified as that Minipterus philipsi. This is this dichotomous key. So they are having the characters, specific characters that bifurcate to and identification characters are there specific. So if that membranous between the elongated that digit 2 to 5 or 4 and these are true flying, so charactera. Then if that membranous weak not absent or digit of limbs normal, then go to second. Then diastema or the space between incisors and molars present, go to 3. And then if diastema absent, go to 4. So if go to 3 that diastema is present, then also bifurcate. The single pair of incisors in upper jaw or ears are normal in rodentia, and two pairs of incisors in upper jaw or ears long, this is lagomorpha. And then come to that fourth point, that diastema absent. We all are insectivores, the snout long or bushy tail in scandens or tree shrew, and that if that snout is short or tail not bushy or the small, if that group is Ellipotaphyla, all insectivores under the Ellipotaphyla. So this dichotomous key, this is primary level dichotomous key. That species level, then that order to that subfamily, then subfamily level, then general level, subgeneral level, tribe level, all up to that species and subspecies level that dichotomous keys are available. Or in other groups also that matrices are also identifiable for the species level identification. So now these methods of that non-invasive methods for that study of smaller mammals. So that some behavioral patterns, population studies, camera trapping, GPS tracking, these all methods are without any sacrifice of the specimens or indirect methods for identification up to species level. So these are the behavioral studies will stay without any specimens sacrifice. So if this is the diurnal roost of this Cerberus medius, you will monitor the behavioral that individuals are migrate in the summer. How many specimens are there? How many individuals are there after migration? How many individuals are staying back? Or when that come back to this roost, these all are behavioral studies will take and will take these smaller mammal studies. So and also a population studies in our that rodents that capture mark recapture method, when you go to field or use that this ceramic trap or other trap, you have to collect the individual live individual and mark that by paint or any rings will fitted to that, and then again left to the field, and another if this individual will trapped in the trap. So you will take observation, make that the trap that earlier that specimen will that captured specimen after that recaptured. So that population study will people will do, and that male, female, or that measurements also just ethnize normal that local ethnize is there, and after the measurements taken, we will free to move in the field that specimen. So no damage to any individual. Then another that camera trapping will be used in the larger mammals, but it's also in the smaller mammals. We will use that the camera position will near to the trunk, lower side up to that near to road and all. So we'll take the capture or that movements of that animal, and will helpful for identification of the specimen. Then this wildlife tracking devices are in nowadays using in the smaller mammals also for that tracking of that animals and all. Then scat identification. That larger mammals, our carnivores, specifically carnivores, predate on that smaller mammals, and that scat. If you collect that scat sample, having that bony part or undigested part or in that hairy part, through that we will identify these lower fauna or a smaller mammal fauna from the scat without the trapping the animal. So they are not disturbed to that smaller mammals or that larger mammals, and will that identify these material through analysis, and the species reported from that fecal matter. So we will know the prey predator relationship or that smaller mammal diversity of that area. So indirectly we will know that diversity of the that smaller mammal without any trapping. So this is a scat of insectivorous. This is a hedgehog scat, and after cleaning, observed that some these mouth parts of these isopterans, that termites are there. If any expert on that termites, we will he will go through the taxonomy and identify that species also. This trichotaxonomy, this identification through hair impression pattern. In the lab, will that take that hairs from the body of that specimen, and through that impression patterns, see this within the genus having different type of patterns are there. This in that Bandicoota indica, that mosaic patterns are there, and in maxima that chevron patterns are there. So these characters are supporting for identification other that morphometry or DNA or all these. This that trichotaxonomy part is supporting to the for identification of the species. Then come to the DNA barcoding. Even that roadkill specimens tissue will decay and identify up to species level. Whenever go to the field, if that roadkill is there, you take the specimen, whole specimen for morphometry. If that specimen is nice for preservation, or unless you will take that tissue from any body of the that specimen and preserved in that 100% or 70% alcohol, and DNA will extract and build a tree and will identify up to species level. In our study, last year we published one paper on that phylogenetic position of that Malabar spinatory mouse, Platycanthus lazarus. We found one specimen killed by that raptor and found in a dead specimen. First time in that barcoding will done on that Malabar spiny tree mouse, and observed that the this Platycanthamidae earlier, that family having two genera. This diploma is reported from China and Vietnam, and that's our Platycanthum is found in that endemic to Western Ghats. But that's the molecular studies and phylogenetic study reports that these population are very much different, and we coined one new family, the Diplomati, for that Chinese and Vietnam population of Diplomais. So now comes to the that owl pellet analysis. What is meant by that owl pellet? So these owls regurgitate undigested food material through mouth in the form of oval and greenish black colored pellets. Depend upon that the size of that owl, that spotted owlet size, the pellet is very much small, or that larger that barn owl and all sizes that owl having larger size pellets. So where they found that pellets, the pellets are generally found near the roosting sites, nests, and also in feeding areas of owls. After collection of that pellets, if you that wash or clean, we found that undigested parts of bony parts of that individual the prey material or prey individual. So this is the one shrew, that Suncus murinus, that swallowed by that brown owl, and this see these bony parts are limb bones, and are that vertebrae are there in all limbs and shoulder bones are there. But for identification of the species, we need the skull or mandibles, because that identification, most of that smaller mammals are important of that dentition or that whole skull measurements. See this that rodent skull material collected from that owl pellet. These are the bandicoots, then Golunda, then ratter species. See how that dentition is different in that cusps are very much distinguished in different genera. See these ratters having different patterns, bandicoots having different patterns, Golunda having different patterns, Tatera having different patterns. See, even if that skull not found, and even that only that molar part also if found in that owl pellets, we will identify up to species level. See the incisors having grooved incisors in Golunda and that Tatera, and these characters, see if the skull found, see this anterior palatal forema, structure of anterior palatal forema is also very much important for identification of the species level. If that molars not found, even that sockets are very identifiable in that genus to genus or species level. These in Bandicoota indica, these structures in the lower molars also there, and this absent in Bandicoota bengalensis. So if only lower moles found, so we'll get distinguished the character is there. So if not there, the Bandicoota indica and Bandicoota bengalensis will identify easily. We that made manuals for the identification of these mammals or the smaller mammals. This is published in 2009. That identification of some small mammal species through all pellet analysis covered all bats, that shrews, rodents. Reported that from that central India to our Pune area and this region Deccan area. But in this manual, later this in 2022, this a manual for identification of prey remains from all pellets. So Mehta madam and all reported that all vertebrates, like amphibians, birds, then the mammals, all will identify up to that bony parts, and that photos are there. Even that insects parts also there. So we'll go through, if you go through this manual, we'll identify, easily identify this that prey atoms found in the owl pellet. What are the advantages of that scat and owl pellet analysis? These samples can be acquired without disturbance to the predator species. Then used for the taxonomic study of prey species, their diversity and distribution, as well as in the studies of predator behavior patterns. Then analysis serves as a non-destructive means of diet determination. That pellet material will go through diet determination to so non-destructive. You will not collect any specimens from live specimens from on the field area. Then that represent a non-invasive sampling techniques which provides a valuable source of DNA for studying population genetics of the prey and predator species. Small mammals are very much important, play important role in the ecosystem, and that they are pollinators, then seed dispersers, and that supports forest regeneration and maintain forest health, aerate soil, that rodents burrows, that they will prepare burrows, so they aerate soil and allow for increased plant diversity also, then control insects, bats control insects, then provide food for other carnivores also. Then conservation implications of small mammals in India. These smaller mammals in India face major conservation challenges driven by the habitat loss, urbanization, and traditional that wild meat hunting. Because conservation efforts typically prioritize that charismatic larger mammals like tigers, elephant, and all. Smaller species like endemic rodents and shrews are that understudy. So, leaving them highly vulnerable to unmonitored population declines and localized extinction. So, for that, this key conservation that implication for smaller mammals will take that that habitat fragmentation and human-dominated landscape. Many smaller mammals rely on that specific microhabitat. The species are threatened by urbanization, tourism, or overgrazing, and that wildfires. Then next to that widespread that hunting and wild meat utilization. So despite legal protections, hunting for subsistence and traditional practices is widespread. Uh, then pesticides and agrochemical use. So smaller mammals are highly sensitive to agriculture runoff. That bioaccumulation of pesticides severely impacts their populations, and that disrupts local food webs. And the community-based conservation, that because that government reserves often lack the resources to monitor or protect smaller species, this community reserves play a critical role, and awareness programs aimed at local communities help halt that retaliatory that killing and change negative perceptions toward the rodents and bats. So that's through that local awareness programs about to protect or monitor on the species reported from their area. Through that Wildlife Protection Act 1972, that now Amendment Act 2022, in Schedule 1 covered Scandensia, one species, that Tupia nicobarica, and 32 species of rodents, then 2 species of Lagomorpha, 2 species of that Lepotapla, and 6 species of that Chiroptera covered under Schedule 1 of this Act. And Scandensia, one species, this Anathena eloti, rodents, 8 species, Lagomorpha, 3 species, Lepotapla, 9 species, and Chiroptera, 6 species covered under Schedule 2 of that Wildlife Protection Act 1972 amendment up to 2022. Uh, through some people, our assays this species level in that ISN categories. One case study I'm talking here. Uh, in 2008, this Kondana rat covered under the critically endangered because only that species reported from that Sinhagat plateau only. After that Samir Bajru study in 2015 found that this species is reported from another three localities like that Torna, Rajgad and Raireshwar. So that occupancy area is more than earlier is 1 square kilometer area occupancy, and after that that three location added, the category we change that cover to endangered. So these type of assessments are repeatedly or that after 10 years will done. So for that the study of the smaller mammals is very much needed. Then challenges in smaller mammalian studies. What are the challenges? Are that lack of that baseline data, the comprehensive distribution maps, the population densities, and basic natural history records are missing for a vast majority of rodent, bats, and shrew species. So because of that lack of that baseline data, we'll not go through that earlier data, and we are for identification of species and analysis is very much needed that baseline data. Then cryptic species, that many smaller mammals look identical externally, requiring complex and expensive molecular or karyotypic analysis for accurate identification. For that, studies will required more collaboration. But if I am only a taxonomy man, only that classical, I use only classical taxonomy. But that for phylogenetic study needs the collaboration of that phylogenetic man. So he will analysis up to this DNA barcoding and all, and the relation of other species. So we'll take more results of that cryptic species and identify the easily identify that cryptic species also. Then we'll found that outdated keys are there. Identification literature is often this old, and that fragmented or inaccessible, or field identification is very difficult through that outdated keys, because this see this uh, as example of rodents, these alarmant trees are there, and at that time that specimen, on the basis of that time specimens found, they the percentage of that hidden body length and tail, and then the skull characters and all is given. Without any that field guide is very much important for without that killing that animal is necessary. Then trapping constraint or logistic problem that needs more number of traps, and it's very difficult to carry without logistic support. Only if I thought, okay, I am going to the field, and that trapping will be done, and that specimen will collect and take without any assistance of this and that logistic support can't do. They need the team of two or three persons for this trapping and all these things. Then priorities to charismatic species like larger mammals. So needs more number of that conservation funding and focus heavily favor large species like tigers, elephant, and other larger mammals, and leaving that smaller mammal that's under study. So needs this priority to these non-charismatic species also. Then permission required from the wildlife authorities, that State Biodiversity Board under the Rules and Regulation of Wildlife Protection Act 1972, and other that Biological Diversity Act 2002 for that surveys and collection of the specimens. And that special care should be taken during that handling the live small mammal specimens, which results of many zoonotic diseases like leptospirosis, plague, that's hantavirus, that rat bite fever. So during the field or handling the specimen, we'll need to use that mask and hand gloves and protect our body from that ectoparasites on that the species found in the that smaller mammals specimen. These are the references. You will go through this for further reading or details. In the last, I acknowledge people that thanks due to the director, Zoological Survey of India, Kolkata, and our officer in charge, headed the Western Regional Center, Pune, for encourage me and providing facilities for the smaller mammal studies. I am also thankful to the authorities of Maharashtra Vriksha Samvardhini, Pune, then Institute of Naturalistic Education and Research, then Department of Botany, RJ College, Mumbai, and Barrister Barasaheb Khardekar College, Vengurla, for inviting me as a resource person. And I also thanks to Sharma sir, then Dr. Vartak sir, and Sri Rajgopal Patil sir for inviting me personally. For further details, you can email me on this ss.talmale@zsi.gov.in anytime, and share your any problems or queries are there. So I will in contact with you by through mobile or WhatsApp, and guide you accordingly. Or you will visit ZSI Pune for any query. Thank you.
Thank you, sir. Thank you so much. Sir, it was quite interesting. Especially, I found it very fascinating that you could identify, there are methodologies now in place to identify just from hair specimens and scats, and those are quite interesting, especially as an amateur naturalist. I think those are something that I feel they are also have a ability to contribute. Um, so do they approach you like, if you mentioned your email, how should, most of the people take photographs when they come across these small mammals. Are these photographs also quite useful? Is it possible to reach a good level of identification based on these photographs? Or what do you suggest, when people take photographs, especially with their mobile phones and stuff like that?
It is very difficult to identify, but bats will identify photographs of the bats. But for that rodents, smaller that mouse and rats and all, without specimens, will difficult to identify of the species and all.
Okay. So I think there has to be something, either a tissue or hair or something, or if you have scats and all, that is another very good area. Do you see, sir, people are aware, or the naturalists are aware of these things, and they come to you often uh, for identification?
Yeah, aware. Most of the time, that owl pellet material received from now, earlier that without that awareness, that only that our area, Pune area, and that central India, we received the specimens or that owl pellet material. But nowadays, receiving from northern India also. So people are now aware about that.
Mm. And sir, I'm sure you must have come across a lot of very interesting cases or specimens and stuff like that when people approach you. Is it possible for you to tell us any such interesting cases that you came across where, or like something where you found very surprising species or something unknown?
Yeah, from see, I've not gone through that. If I gone through that, the Himalayan range, that holes and all these things. But whenever I received that pellet material from that area, it's very much interesting for me to go through only based on that dental part and all. Identification is very difficult, but this literature is on the basis of literature will identify up to that species level.
I think, sir, it is inspiring if we, this is a very good pointer, like people who go out in nature, who travel in different places. they can collect pellets of birds like owls and raptors, or they can also collect scats, like you mentioned.
Yeah.
And today, those will be quite useful to understand their ecology or their behavior. What kind of food they're eating, what kind of small mammals are present in that area. I think that will be quite interesting. So,
Be- Because nowadays, sir, it's difficult to that collect that specimens. There's a rules and regulation is other side, but in the field also that not a single day program is there. If you collect, you want to collect the specimens from any area, you want to that arrange the traps and all repetitively,
right.
because for rodent studies, all that we, for first day or second day will not attract that. Bait and all these things. They are
Right.
aware anything is there. So that shyness is also there.
Right.
So for long-term studies, you need 10 days or 20 days of that field study. So we will get that specimen or and also difficult to get that if you are want that any specific species. This is very difficult. If I trapped a specimen and will kill and see, array, yeah, this is the only ratus species. I want another species. So this is very difficult. Any that targeted species.
Right. Right. Before we take questions from others, first I would like to request Sharma sir to give his comments, sir. If you want to add something, sir.
Well, I was a part of the organization, so I know the difficulties and even advantages rather of collecting these small mammals, because I myself has collected from different areas from Kerala and all, and they turn out to be the new records for India when Dr. Pradhan was alive. And of course, it's a very interesting group to study, and not interesting, but it's difficult also, because you have to undergo lots of complexities like skinning, then taking out skull measurement. I mean, it's not that only. You observe something under microscope and you come to know the species. It's very, uh, I mean, complex. So, but very interesting.
Yes. Yes. Thank you, sir. Uh, Thank you. Upadhyay madam, Vartak sir, would you like to comment or add something?
Uh, it's very informative lecture actually, because we are knowing plants more than that of these type of animals, and that too for the mammals it is very interesting.
Right.
Just I want to ask that if there is any effect of the temperature and humidity, because we always uh, speak out that the Pune is a very good climate for the
Yes.
preservation of such specimens, but I don't know about the mammals. So if we can add a little, uh, which which what actually exactly the temperatures and what variations actually such type of specimens can withstand. I think that because once people know to collect the specimens, they just collect, but they don't have that idea how to carry that specimen
Right.
up to the expert person. But it's a very informative and very good lecture. It's a very good addition in on our mission there.
Thank you, Upadhyay madam. Talmale sir, so from preservation perspective, as Upadhyay madam asking, are these all many of these specimens available for people to see in Akurdi or any other
Yes,
ZSI?
yes, yes, yes.
Yes.
All that dry specimens and also that in wet preserved specimens both. But
Okay.
nowadays that stop this dry skin preservation because no taxidermist is in the office,
Okay.
and is also difficult to prepare in the field also. So will directly preserved in alcohol. So
Okay.
most of the specimens nowadays will preserved in the alcohol.
Okay. But do
Thank
you
you,
need?
sir.
Thank you.
Hmm.
Do you need any climate control for these preservations or?
No, 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 no. That's Pune area is very much
Right.
known.
Yes,
Yeah,
as Upadhyay madam mentioned. Yes, thank you,
yeah,
sir.
yeah.
Sir, before we go for further questions, I would like to request Awad sir to do the vote of thanks, and we will quickly take, there are a couple of them in the chat box, and we will take any individual questions as well. Sir, I hope you have a few minutes, so uh, I request you to stay on. Awad sir, over to you, sir.
Yes, yes, sir. Yes. Yeah. Good evening, everyone. As we come to the end of today's enriching session on understanding India's small mammals, the identification, methodology, and conservation, it is my pleasant duty to propose the vote of thanks on behalf of Maharashtra Vriksha Samvardhini Pune, Institute of Natural History Education and Research, Balasaheb Khardekar College, Veggurla, and Department of Botany, Ram Niranjan Jhunjhunwala College, Mumbai. I extend our heartfelt gratitude to our distinguished speaker, Dr. Shamkant Talmale from Zoological Survey of India. Sir, thank you for sharing your vast knowledge and especially the field experience on India's fascinating small mammals. Your insightful presentation on their identification, research methodology, ecological significance, and the conservation challenges has opened our eyes to a group of animals that are often remain unnoticed but play an indispensable role in maintaining the ecosystem balance. We are truly grateful to your time, expertise, and the interaction with the participants. I express my sincere appreciation to our collaborating organizations for coming together to organize this valuable academic program. As Madam mentioned, this is a very good value addition and for promoting the awareness about biodiversity conservation. My heartfelt thanks also to all the faculty members, researchers, students, nature enthusiasts, and participants who joined us today. Your enthusiastic participation throughout the thoughtful questions and the active engagement made this session lively and meaningful. A special word of appreciation goes to the organizing committee and the technical team for their meticulous planning and seamless management for this online event. Their dedicated efforts behind the scenes ensured the smooth conduct of today's program. As we conclude, let us remember that conserving biodiversity begins with understanding every component of nature, however small it is, the smallest mammals that quietly support our ecosystem. May today's session inspire us all to observe, to appreciate, and protect the remarkable wildlife around us. Once again, I thank Dr. Talmale and thank you everyone for your presence and support. I am putting a link to the WhatsApp group in the chat box so that every month at least 3 to 4 such meaningful lectures, which are organized by these 4 institutes collaboratively, can be attended online. The recordings of the same are also available on Mission Devrai channel, YouTube channel, so that you can go through the archives and visit all these sessions once again. Thank you, one and all.
Thank you very much, sir.
Thank you so much, Awad sir. I think you captured the entire essence in such a beautiful way. Thank you so much. Talmale sir, I think there is one question. Anybody else who has a question, you can raise your hand in Zoom, or you can put it in the chat box. There is one question. Nilesh Chavan is curious about the number of species of porcupines. So I think he is quite interested in India. Do we have, how many species of porcupine do you find here?
Three species are
Okay.
three species reported from India.
Sir, are these all like you mentioned about DNA barcoding? So do we have a good coverage of DNA barcoding in our small mammals, or do we still need? tissues or samples or those things to build a good database of DNA barcoding. What
Yeah.
and is this something that people can approach you for, or how should they go about with this DNA barcoding identification?
Yeah, yes, need because in gen banks, very few that data is available. So needed all database and all. So in future we'll build larger trees and all these things. So
Right.
for better results. So anybody collaborate, you want to collaborate, he will contact to our laboratory and no problem for that.
Okay. Wonderful.
Yes.
Uh, Thank you so much, sir. Uh, That Kondana rat also that you mentioned, that was also quite a very interesting case of distribution. Sir, is that rat something you need specimens, or is that something we can photograph in the field? Because it looks like very close to Pune, that range of this very unique species.
Yeah, people are doing working on that population studies and all.
Okay.
Yeah, yeah.
Okay. Great.
People are doing.
Okay.
Hello, sir.
Right.
Can I ask question? Sanika Gupta here from Junjunwala College.
Yeah,
Yes, madam.
please go ahead.
Yes.
Recently I came across an article where they have stated rare giant flying squirrel, which is native of Western Ghat, finds its home in Chhattisgarh Reserve Forest. So is it a good sign? Udanti Sena Nadi National Park.
Okay.
It was frequently now sighted over there. Udanti Sita Nadi Tiger Reserve from Chhattisgarh.
Yeah, yeah.
So,
That giant squirrel, Rotva indica centralis, reported from that area, Chhattisgarh and Odisha area.
so their range is expanded or what?
No,
Earlier,
no,
it
no.
was,
That earlier people will not identify or that area or any reports, but that species is reported up to that Odisha and all.
okay. And sir, one more question. If we want to bring our MSc student to ZSI visit, what is the formalities we need to do?
Email sent to that our office email ID wrc@jsi.gov.in. So that will reply to you, and you will arrange that trip to our museum.
Okay.
Hmm.
You, sir. And when I was at Modern College Pune, MSc, Dr. Ghate sir used to bring us to Poud Road Institute. That time it was at Poud Road.
Yeah,
So
yeah,
long, long back. So I would like to take my student to Akurdi.
yeah. And then near to Akurudi railway station, so no that transport problems
Yeah,
and
I
all.
had been to there also, sir, once when we were at MSc.
Yeah, so okay,
Back, long back.
okay,
Surely,
okay.
sir, I will get back.
Yeah. No problem.
Thank you, sir. Thank you.
Thank you very
Thank
much.
you. Thank you. Folks, there is a link. If you are not part of our WhatsApp group, Awad sir has put a link in the chat box. So do join and you'll come to know of our future talks. There are ones which are pretty regularly organized. So do
Rajgopal
join.
sir, I think
Yes, sir.
Savarkar sir is also there. You
Oh,
can
yes. Savarkar sir, okay. I thought I saw him earlier. Savarkar sir, would request you to add your comments as well. We'll sincerely appreciate that.
I think this was a very illuminating presentation. Small mammals, as it was said right in the beginning, are neglected. And by and large, the knowledge is quite poor. So I think by that uh, uh, yardstick, I would say that this particular presentation covered everything. And there was the scientific method involved in taxonomic classification important thing about distribution of species and non-invasive methods that are there. I remember I was as a greenhorn, I was in Melghat Tiger Reserve in 1973 to 79. At that time, we had collected scats from various places, and I think coming back to this particular presentation, we had quite a good collection of scats of I mean the pellets of owls, and we had Vidarbha University in Amravati, where there was Dr. Subnis from the zoology department. And he used to come and identify the contents of those things. And I think the importance of scientific methods started right there. And there was Dr. Khajuria,
Yes.
a famous bat
Yes.
specialist. And not just bats, but he was a first-rate field person. He used to come from Jabalpur. He used to visit our tiger reserve in Melghat. And we learned quite a bit from him. And as I was looking at the bat, various pictures of bats, his image just came to my mind. And I think by and large, this is an important thing that my colleagues from forestry department, especially those who are working in the wildlife wings, this is an important, very important presentation. Even if they find it difficult to get into the scientific methods, but the importance of it, and I would say the role of these small mammals. We always call forests as naturally self-regenerating entities. But what makes it possible? Small mammals play a very important role in that, as this presentation has made a reference. So I think by that, I really appreciate this presentation. Thank you very much.
Thank you very much, sir. Thank you.
Savarkar sir, you always bring out all these wonderful angles to look at these things, and that is also very interesting every time. And especially you have seen such a big span of our country going through different phases of wildlife conservation, and people like Dr. Khajuria that you mentioned. I think a book from you is due, sir. All these got to be somewhere, and it is always wonderful to hear from you about these things.
Thank you. I'm certainly, I'm making an effort, and the experiences I got, basically ecological, management oriented, related to, of course, wild species and people, the impacts of development and things like that. And I was very fortunate to travel right from the northeast, northwest, and down to Kanyakumari, and visit a very large number of areas. So there are some interesting things. So I'm making an effort to put those things down, and I hope they would see the light of the day. Thank you very much.
Thank you, sir. We eagerly look forward to that very much, sir. Talmale sir, there is one question from Irfan Ramiz. He would like to know, how can a casual observer distinguish between the different classifications of small mammals without the help of any equipment or measurement?
Yes,
What level can they go, sir?
yes, yes. That field guides are available, and that squirrels like three-striped squirrel, five-striped squirrels will be easily identifiable. Then giant squirrels also is it. That problems in that smaller means that mouse and rats having identification is very much difficult. So for that, you should know that needs that one specimen from that area for identification. So without that, other that bats also that field guides are available. So on photographic base also we will identify up to a genus level or up to species level also in some genera. So that is not a problem, but for some smaller groups like mouse or that some shrews mostly needs at least one specimen for identification and for the tissue needs for the DNA barcoding and all phylogenetic studies.
So, is there any, is it legal for people? I mean, if we find a dead specimen, that is fine. But you mentioned about the conservation status of and the protection status. Many of them don't seem to have any, they don't seem to be in a schedule where they are well protected right now. So I guess the data needs to be there to be able to protect them, right?
Yes, yes. Even some rat species also in the Schedule 1 and Schedule 2 species.
Okay.
And collection from that protected area is very much difficult.
Right.
From
Yes.
urban area, there is no problem because people are they use that pesticides and all, rodenticides and all. No, there is no problem. But collection from the protected area is very much difficult. Without permission and from authorities, it's very difficult.
But I think
I also take permission from the authorities.
yes.
Even we also entered that any protected area without that permission we got entered.
So I guess meanwhile, people who are listening to this or listen to this on YouTube, they can go out to look for any possible roadkills or other dead specimens or scats or pellets, owl pellets. Those will be a good source to get some good level of data, which is desperately needed for this group of animals.
Yeah, roadkill with all coordinates data, locality details, because that distribution of that specimens or species is very much important. If any rat species is there, if that specimens from southern area or western Ghats, southern western Ghats, maybe that species is different from our area.
Right.
So there's coordinates and that locality is very much important.
Right. Thank you so much, sir. Thank you once again.
Thank you.
Thank
you
you,
very much.
everyone. I think we can end this call here. Thanks all for joining again. Thank you all.
Yes, thank you, sir. Thank you.
Thank you very much.